And we're back with DNA replication, the highly anticipated sequel to the first mini lecture, Intro to DNA Replication. But before we start, here are some random fun facts about the human genome. The human genome has more than 3 million base pairs per haploid set of chromosomes. That means right after S phase and DNA replication, there are 6 billion base pairs in a diploid cell. The rate of replication is 100 nucleotide bases per second, so really fast. Okay, now to get to the nitty-gritty of DNA replication. During DNA replication, each of the two strands that make up the double helix, so this top one, this one here, and this one here, serve as a template for which new strands are copied. The new strands will be complementary to the original strands. When two daughter DNA copies are formed, they have the same sequence and are divided equally into the two daughter cells after mitosis. Before replication can start, the DNA has to be made available as a template. So these histones, which I hope you remember, are responsible for coiling up the long DNA strand into this tightly condensed X formation. The histones must be removed, and once they're removed, then the DNA can unwind. At the origin of replication, which is right about in this area, just upstream of the replication fork, a pre-replication complex is made with other initiator proteins, and then helicase and other proteins are recruited to start the replication process. Helicase uses energy from ATP hydrolysis to open up the DNA double helix, making these two original strands available. Replication forks are formed at each origin of replication as the DNA unwinds. The opening of the DNA double helix causes an overwinding or kind of a supercoiling of DNA ahead of the replication fork. This can be resolved with by this enzyme called topoisomerase, which kind of reverses that coiling to keep the DNA from supercoiling all over again. An enzyme called primase, which is not shown here but would be working right in front of the DNA polymerase. This primase lays down these nucleotide primers, which are basically little templates that the DNA polymerase can turn into nucleotide bases. Although only one is visible in this figure here and here, there are actually 3 major DNA polymerases. Polymerase 1 and 2 work on the leading strand, developing the daughter strand from the leading strand of DNA, and polymerase 3 works on the lagging strand, helping that daughter strand replicate DNA. So let's look at the leading and lagging strands to understand why they are labeled that way. DNA polymerase reads the primer and synthesis of the new strand always from 5' to 3'. That means that on the top strand, it's reading from right to left, and on the bottom strand, it's reading from left, the 5' left end to the right. Now look at this top strand. We can see that topoisomerase is stabilizing, helicase is unwinding and opening, and then these two components are working hard just up in front of the DNA polymerase enzyme. On the bottom strand, the 5' to 3' direction is left to right, and since the origin of replication is at the 5' end right here, DNA polymerase can only lay down nucleotides in short fragments, which are called Okazaki fragments, while it waits on more of the DNA to be opened so it can keep going. So the DNA polymerase kind of jumps along the bottom strand, replicating 5' to 3' in short fragments. Because of this, the bottom strand or the lagging strand will always be a little bit behind the leading strand. This is why the top one is called the leading strand and the bottom one is called the lagging strand. The Okazaki fragments in the lagging strand are joined together after the DNA polymerase goes by. They're joined together, and the gaps that are remained are sealed by an enzyme called DNA ligase. which forms phosphodiester bonds. Let's take another look at how these Okazaki fragments are formed. So here we have the leading strand, DNA polymerase moving from right to left, 5' to 5' to 3' on the original strand, and 5' to 3' on the original lagging strand. All right, so DNA polymerase is moving the replication from 5' to 3' on the leading strand, going this way, and we see the daughter strand being built complementary to the leading strand and moving this way on the lagging strand, with the Okazaki daughter fragments going still from 3' to 5'. Since DNA polymerase works behind, so DNA polymerase would be here and here, since it's working behind or downstream of the origin of replication, which would be in this area, DNA polymerase can only work so far before it has to wait for another segment. After replication, the gaps between the Okazaki fragments and the lagging strand are sealed by DNA ligase.